So hello and welcome to another class of abmebiosciences. This is Abhishek with you. So today I'll be talking about mechanism of DNA ligase, as you have learned from my thumbnail. So I've seen many videos in YouTube and those are not so informative or not so perfect in my knowledge. That's why I've created this video for you guys to clear your knowledge and the concept, where it is and how it is happening. So coming to DNA ligase. So DNA ligase, you know that two types of DNA ligase are there depending upon which energy source are they are using. So if you are isolating the DNA ligase from eukaryotes or viruses, that time it will be using ATP as the energy source. The enzyme will use ATP to seal the nick. So the function of DNA ligase is to seal the nick created during DNA replication. Okay, so that nick is particularly sealed by DNA ligase enzymes, and that DNA ligase enzymes can use two types of energy source, that is ATP or NAD. So if that is from eukaryotes or viruses, that enzyme will be using ATP, and if it is from the bacteria or bacteria is using that enzyme, that time it will be using, for bacteria it is NAD. So this is the basic difference between two types of DNA ligase enzymes. Now how it's happening, what is the actual mechanism of DNA ligation or sealing the nick, that I'm going to discuss here. So first you see this structure here. This is the enzyme structure I have drawn. Already to save the time and make you understand clearly as soon as possible. So here this is the enzyme complex, or you can say the DNA ligase. Okay, now the first step of sealing the nick is it has to be AMP acylated, means the adenosine monophosphate from the ATP or the NAD has to be transferred from the ATP or NAD to the enzyme. So the first step is AMP acylation. So here this is the AMP form. Okay, now the R group here, it denotes PPI in case of ATP, and if you are taking NAD, then it is denoting NMN in case of NAD. Okay, so the R group denotes either of PPI or NMN. Okay, and the AMP group is denoted by this phosphate, ribose and adenine. Now this is our substrate or group of interest. AMP is now in the second step, that AMP is added to the serine residue of the ligase enzyme. So the serine residue present in the DNA ligase enzyme is being attached with or acylated with the AMP group that is drawn from where, that is ATP or NAD, okay, depending upon the enzyme. Now here our enzyme is now ready and attached with the AMP group. This is the first step of DNA ligase reaction. Now you have to have a substrate to seal the nick. So here our substrate is this DNA, which is broken here, or there is a nick. So if it is 3' to 5' first strand, second strand is 5' to 3'. So here 5' to 3' you can see this is the hydroxyl group, and if you see this is the 5' end, then there the phosphate group is hanging around. So this phosphate group has to be activated before or to seal the nick. So the activation is very, very important, and that activation is done by the DNA ligase enzyme by first accompanying the AMP group with it and then transfer it. So now the enzyme DNA ligase is now moving out, and that group here, I'll show you in the red form, and this group is going to be transferred to this phosphate, and that is the activation stage. So this is the step 2 or activation of the phosphate or 5' phosphate group. So if I draw it here, it will be like this. Okay, and here it is ribose and adenine. So now this group is transferred to the phosphate, which is present in the sealed, that is the nick present, not sealed yet, it is not sealed. So now it is activated, the 5' phosphate is activated with the addition of AMP that is transferred from the enzyme AMP, and the enzyme is out, and now that has transferred its AMP form to the 5' phosphate or the phosphate group. So the phosphate is now activated. Here again the ligase, now what will happen, here you can see all the steps. The one removal or displacement of a phosphate is adding or creating another ester bond. So here the one important thing after this transfer of this phosphate group, the AMP group to the phosphate or the activation of 5' phosphate, is that after the activation of this phosphate group, here this hydroxyl group will attack the phosphate group or this bond, and it will be the reason from the displacement or the displacement of the AMP group from the nick site, and the nick is sealed with a phosphodiester bond. So the nucleophilic attack of the 3' hydroxyl to the 5' phosphate group is going to happen only when it is activated with the transfer of AMP from the enzyme to the that is your substrate DNA. So this is very important. In the next step, that is the third step, this phosphate AMP group will be removed. Again, I am not writing the total structure. So this AMP is now out with the help of DNA ligase, and after that the nick is sealed. So now you have a sealed DNA, that is fully functional DNA, which can be again replicated or transcribed. Okay, it is ready for transcription. So this is the mechanism of DNA ligase, which is very easy, but you have to understand that the crucial stages are activation or the attachment of the AMP group to the enzyme, the first step. Second one is the AMP group is again going to be transferred from the enzyme or by the enzyme to the phosphate group that is present in the nick, that is the 5' phosphate group present in the nick, and that after activation it will be or the nick will be sealed down after the exclusion of the AMP from the sealed site or the nicked site. So after the exclusion of the AMP phosphate from the nick, the nick will be sealed. So this is the basic mechanism of DNA ligase reaction. I think it's a very useful class for you. So if you like my classes, don't forget to subscribe and come back again. So thank you. So if you like my classes, give a big thumbs up and don't forget to subscribe my channel because these things stimulate me to take futuristic classes for you guys. So thank you and be with me.